Hi, everybody. Welcome. We'll give people another couple minutes to join, and then we'll get started. Klaus, you seeing my screen on your end?
Yes, thanks.
Perfect. How's your day been going so far, besides the hot weather?
Very good, thank you. Busy times. We just had a German medical image computing conference here in Heidelberg, which I was responsible organizing. So 3 days of many people running around here, interesting conversations, and it was great. BVM 2022 it was.
Nice. Yeah, too bad I missed that one. Maybe next time. It does sounds pretty busy.
Yeah, it's really nice. Great community.
Mm-hmm. Yeah. Okay, so I think we got a fair amount of people already, so let's get started. So welcome everybody to, we still call this Bay Vision a virtual meetup, but it's really, a global virtual meetup. We try to bring in, the best researchers from the medical computer vision community to this meetup. So today our lecturer will be Professor Klaus Maier-Hein. We'll talk about machine learning and medical imaging, current challenges. Clinical impact has been rather modest. So there's so much research going on, both in academia and in industry. I'm in London for the Hamlin Symposium, and you're hearing and seeing amazing things, not only on the robotics side, which is the main topic of this conference, but also on the imaging side, and all the robotics solutions need imaging. So there's so much going on sort of under the hood. But what are really the obstacles for putting all these amazing technologies into clinical use? Klaus will be sharing some of his insights and some of his research on those real world challenges for actually bring benefits in practice in the clinical setting. Brief word about us. Just a minute. Excuse me. I'll go back to the slideshow and I'm going to share it again. So we are RSIP Vision. We're holding this webinar as a service to the community. We do visual intelligence solutions for medtech, dealing with three main areas: planning, surgical planning. So not only, for instance, segmenting CT scans, but also elevating modalities such as X-rays and MRI, and taking those to the next level to enable next generation surgical planning. Intraop navigation, such as a virtual ruler using surgical tools, 3D reconstruction of scenes, for instance, for judging completeness of mapping of tubular structures, and of course the analytics that go around that. Our company provides differentiating technologies to global medtech industry leaders, and our solutions are deployed in medical devices and used in thousands of clinical procedures. And we, of course, are very, very interested and eager always to learn about the new research that comes out of the leading universities in the world, which is why we have organized this webinar and bringing speakers such as today. So without further ado, I will introduce our speaker. Professor Klaus Maier-Hein is at Heidelberg University and Managing Director of Data Science and Digital Oncology. He heads the Division of Medical Image Computing and Pattern Analysis Learning Group at Heidelberg University Hospital. His research is focused on deep learning methodology for medical imaging, and also developing the software infrastructure for efficient and effective translation of the results into the actual clinical practice. So Klaus, very happy that you're here today. I will make you the presenter, and yeah, take it away.
All right. Thanks a lot for the kind introduction. Can you see my screen, yeah?
Yeah, all good.
Okay, great.
One more, sorry to interrupt you, one more thing I forgot to mention. So the audience is invited to write in your questions. The webinar interface should let you type in your questions, and then we'll read off some of them to Klaus at the end of the talk, so you have a chance to ask questions of the speaker during this, at the end of this webinar, and of course can correspond with either of us by email afterwards. Okay, go ahead.
Sure, thanks. Yeah, looking forward to your questions. Hi, my name is Klaus Maier-Hein, and I thought about talking about current challenges today. So I wanted to highlight some of the research that we've been performing in machine learning and medical imaging, but also stress some of the obstacles that we are basically seeing. And I want to start my presentation with an example. And you all have heard of transformers, which are really, really successful in other domains, natural language processing, and also computer vision. And of course, people have adopted transformers also for medical image computing and especially medical image segmentation. And here you can see an overview of 4 papers that we were interested in that have shown success in applying transformers to medical image segmentation. And yeah, so one thing that I wanted to show you first is a little overview of results that have been achieved on basis of these papers in the literature. So you can see the unit R model, and you can see different average DICE scores that were published in different papers. On the top here and the bottom, you can see the same for the trans-BDS model. And what you can see is that these results are varying quite a lot. So replication, this shows that replication is difficult. Also, we saw that evaluation schemes between different papers vary quite a bit, and that we also have here different variants of the same data set, leading to those varying results. So we were really interested in testing the potential of transformers. And so what we did is we looked at the most well-known transformer architectures. You can put the transformer in the stem of your architecture. You can also put it in the bottleneck, or you can have a full transformer architecture. And on each of these variants, we bypassed the part that is actually doing the work for the transformer. So we replaced the transformer block with a single linear projection layer without introducing any other convolutions or self-attention operations. And what we found is shown here. So for different transformer variants, we compared the results they would achieve on 3 different data sets here: KIDS 21, BREDS 21, and multi-organ segmentation. And what you can see in comparison to NN unit is that some of them achieve comparable results. So for example, here the unit R and the trans-BTS model on the BREDS data set are quite competitive. However, if you now look at the thin bar, so the thick bar is the transformer model including transformers, and the thin bar is showing the performance of the transformer model without the transformer block. So you can see that basically, yeah, if you compare them, it's not really effective, right? The transformer does not really contribute a lot to the performance here in all 3 tasks in the trans-BTS model. And at unit R, also for the BREDS data set, there's also not much performance contributed by the transformer, which is shown by equivalent performance if we take out the transformer. On other tasks and other models, it seems to be effective, but these are all tasks where we have performances that are far away from the NN unit performance. So I think this shows a couple of things, and it's just my example to start this talk, right? So I think we should very carefully think about our baselines, and these are essential in studies. And this also shows that transformers do not always contribute to the net performance of a model. It also shows that transformers performance are very task dependent, so we need to evaluate our models on different tasks. Yeah, and it also shows that we need to kind of incentivize the use of the transformer block for the transformer, so not to bypass it and use only the convolutions to do its tasks. Yeah, so these are a couple of take-home messages already, but of course my talk has a little bit of a broader scope, and I really want to zoom into some of these problems that I mentioned a little bit further. And I do have the feeling that we are often rushing for better benchmarks, and sometimes this happens at the cost of analysis. So I think it's really important to choose proper baselines. It's really important to analyze the effects that we are seeing, for example, by using ablation studies or anything comparable. Yeah, and it's also very important, of course, to stick to comparable measures, right? Using similar evaluation schemes and data sets. So my talk is going to be structured into 4 sections. I'm going to talk about the data, which is one important bottleneck, I think. I'm going to talk about solving the right problem. Also going to talk about the generalization of results or methods to other data sets and so forth. And also going to talk about the representations that we are learning. So let me start with the first section here. Data. And actually, it's quite interesting. So a large part of my group is currently working on ways to make accessible data that we need to develop our methods. And so one basic technology that we are developing here is the Kapana framework, which aims at allowing medical image computing at scale. So at the moment, as you can see here on the left, a majority of data is not available for AI. And this has many reasons, technical reasons, organizational reasons, and there are also ethical obstacles. And yeah, with my team and with the Kapana toolkit, we are trying to basically facilitate sharing of data. We're also trying to leverage data which is not public. So we are trying to basically enable federated scenarios in order to resolve the major obstacles that we are looking at in the moment, and to make federated data sets act like they were just one joint repository of high quality annotated data sets. So the concept that is followed by this Kapana technology is basically that we directly integrate into the clinics and we interface the often very heterogeneous landscapes that we are finding there. So it's quite complex infrastructure in order to do so. And once we basically are successful in linking to this infrastructure, we are trying, of course, not only to get the data out and send it somewhere, but also to bring the algorithm to the data by using mechanisms like federated learning or also federated validation. And this is all open source. We are using a lot of current technologies like Kubernetes, Docker Swarm, microservices, and I see these technologies going towards like building an operating system for clinics, basically, that we can access and that we can use in order to perform research on basis of it. So we are involved in various running initiatives where we apply Kapana. So these are EU-wide, but these are also national initiatives. For example, the Raccoon network is a network where we are interlinking at the moment all 36 university clinics in Germany in order to exchange information, data, and also algorithms in context of the COVID pandemic. And yeah, basically Kapana solves questions not only for data sharing, but also algorithm sharing. As I said, it also helps workflow integration. It's enables broad validation basically of methods that we are developing. And it's all made for scalability of our results and scalability of our processing and translating basically our methods in practice. One thing that I wanted to show is also the Federated Tumor Segmentation Challenge, which we organized in 2021, the first time. So this is basically the first truly federated challenge at MICAI. So here participants submit their brain tumor segmentation algorithm, which is then sent around the world and evaluated on many, many data sets in a federated scenario. So in case you are interested in the BRADS challenge, for example, and have a plan to submit to the BRADS challenge, you might also consider to submit to the federated tumor segmentation challenge, which is also going to be hosted by MICAI in this year, 2022. You can just submit basically your brain tumor segmentation algorithm and see how it performs on various locations in the world. So if you're interested in this technology, you can check out our website or other channels that we are using here to distribute information about GAP-HANA. Okay, so that was the first challenge. So I think if we manage to just increase our amounts of data that we have available for training and also for testing our algorithms, that's already going to solve a whole lot of problems. But of course, it's not all that we need. So we need to make sure that we are also solving the correct problem, right? So that's basically the aim of our work, to solve problems. But what problem are we often solving? So let's take a look. So here you can see a histogram of different metrics, and you can see that DICE similarity coefficient is, if you look across challenges, by far the most used metric. And it's basically an overlap measure. And it's well known that there are several pitfalls related to the DICE. So, for example, as you can see here, it's on the top left, the effect of small structures, which can be very hard to basically see if you only look at the metrics. You can see the effect of over and under segmentation, top right, bottom left. You can see the effect of shape unawareness. So the metric is basically unaware of the shape of the segmentation that is evaluated. And you can see in D, and this is something I want to focus on further now, the effect of a task metric mismatch. So here you have 4 different regions of interest, and you can see that prediction 1 in green and prediction 2 in orange basically deliver similar DICE scores, but even the green one delivers a higher DICE score. But medically spoken, it would be much better to receive prediction 2 because it includes all the objects that are of interest here. So I've already talked about brain tumor segmentation and the federated challenge that we are hosting at this year's MICAI. And brain tumor segmentation is really a very long-standing problem of the community. So people are working on that for many, many years. I think BRADS is one of the first challenges at MICAI. It has more than 200 teams participating at the moment. So it's really many, many people working at brain tumor segmentation. And what I see every year is, okay, they're increasing the data set size. Methods are getting more and more fancy. And the DICE scores are also increasing and increasing a little bit, not much each year, but they are increasing. So I think many people would agree if I said, okay, this is basically a problem which we have been working a long time on and which is solved quite nicely. And here you can see a typical comparison of a ground truth and a prediction, and you would say, wow, yeah, it looks nice. And the voxel-based metric would say, okay, 94, 0.94, so it looks nice. And now at the same data set, on another slice, you can see that here in the ground truth, you have a small spot of a second instance here on the bottom left, which was not found in the prediction. So this hardly changes anything at voxel level, but it changes everything basically if you look at the instance level. So the instance metric would here be 0.5 because you've only found half of the structures that are of interest. And this is highly relevant. So this is not really good, right? So let's look at this a little more systematically. So what you can see here is you can see a plot where I show the metric result only for images with a certain number of instances. So on the left of this graph, you can see there are here are images with only one instance, and you can see in this setting a voxel level score and an instance level score are basically telling the same story. So we have high numbers here. But as soon as you go to 2 or more instances, the voxel level and instance level evaluation deviate a lot. And you can see here in the histogram, which is shown in red, that many of the cases are affected by this. So it's not only rare cases which have a couple of instances in the image, but it's many images that are affected. So that tells us that voxel-based metrics overestimate instance performance. And so here I show a plot, and you can see the relationship between the DICE score and the F1 score. And you can see that there's hardly any relationship between those two scores, one coming from voxel-based analysis and one from object-based or instance-based analysis. And the similar graph is shown here, the relationship between the Hausdorff distance and the F1 score. And again, you can see there's hardly any relationship. So typical challenge metrics, DICE, don't reflect the detection performance. So we have to ask ourselves, are we solving the wrong problem here, right? And let me show you what I think. So I think that in the clinic, we should always ask, what is the consequence of our finding? And one very important question here is, do we have any progress? So a patient comes to the doctor, comes again, and the question is there any progress? So in our database, and it's quite a good database of many hundred cases, so we can see a volume progress in 502 cases. And if you compare the ground truth with a prediction, we see that we can basically find this volume progress in 87% of the cases, which is quite good, I think. But now if you go one level further and you look at the instance progress, and this is medically and clinically much more relevant, right? Because if there's a new instance, like a new site where tumor is occurring, that is often highly relevant for making decisions on the care. And here you can see that from the 525 cases, we can only find 267, so which is half of the cases, right? And this is really bad, I think. And if you go one step further and we look at the clinically highly relevant case of progress, where you have one instance in the beginning, and then you have a progress to many instances later on, we have 200 cases in the database, and current segmentation algorithms can only find 88 of such cases, which is 44%, less than half. So this medically highly relevant event of progress is not detected in the majority of scans, even though this is state-of-the-art database here and state-of-the-art methods. So there's a lot of work to do. So take-home message here is be aware of and solve the right problem. And we are often tackling problems using semantic segmentation algorithms, which are shown in yellow here. And if you only look at the left column of the medical example, it's a really nice visualization here from Lena Maier-Hein and Annika Reinke in their Metrics Loaded paper. So you can see that semantic segmentation will give you one information per voxel, right? And often we are interested in information per object. And if we want to have that information per object, we need to go to other methods and other metrics. Object detection will deliver one measure per object. And there are methods for instance segmentation where you even get object-based measures, and at the same time segmentation of the instances. So having talked about now years of looking only at DICE score and having talked about the problems related to this, the question is, how can I come from my data set or from my task, from the biomedical question, how can I come to the best metric? And there's an epic effort that is currently being done here, also by Lena Maier-Hein, led by Lena Maier-Hein, of course, including many, many other people, which I'll show in a minute, which exactly this consortium exactly tries to clarify this. So how do I come from my biomedical question to the recommended metric? So many, many institutes around the world are taking part here, all the experts and bright minds. And at the moment, we are in the state of collecting feedback. So it's your chance to also provide your use case and get some feedback on what metric would be optimal. And vice versa, we would get some feedback on how the current process works and what we can maybe improve on the recommendations. So this large-scale consortium is trying to find one recommendation that everybody can agree on how to find the best metric for your problem. So check out this survey here, take part, and provide your use case if you are interested. Okay, so that was the second aspect of the key I wanted to show. Now comes the third, generalization. Yeah, so everybody knows that. You might be interested in a method. You find very nice results from a paper, and then you implement it. And on your own data set, it just doesn't perform as well as you thought. And this happened very often to us also. I showed you an example in the beginning where we had this problem. And I think one very important aspect to generalization from one data set to the other is the hyperparameter tuning. So how does a typical development life cycle of a deep learning approach work? We have a data set. We configure our method using some hyperparameters. We go on, we train our method. We evaluate it using some kind of metric. And then comes the point where we decide, okay, this is my result now, but can I do better? And the question is, what do I need to change on my hyperparameter settings in order to get better? And then once you have your changes, you loop, you train again, you evaluate, and you go back to your hyperparameters. And this complete cycle here is really difficult to perform. So it requires expert knowledge. And if you don't have expert knowledge, it requires a lot of trial and error. And yeah, even for experts, it requires a lot of trial and error because there's no formalized knowledge about this process. So there's a lot of feeling involved, a lot of experience. It's really time consuming and it uses a lot of hardware resources. And I think that the missing standardization in that process really hampers our research progress because we have one paper saying that works, but if you transfer the method to the other data set, it's really hard to get it running, really hard to show that it works also on other data sets, and also unclear whether it works at all. So this is what we looked at when we worked on NN-UNet, which is the self-configuring method for deep learning-based biomedical image segmentation. So NN-UNet is basically exactly targeting that cycle where hyperparameters are tuned and trying to automate that. And in the paper, you can find this figure here, which shows as an example the 100 competitors of the KIDS 2019 challenge. And so what we wanted to see here is in that development cycle, you tune your hyperparameters. And the question is, can I maybe learn from other publications what are good hyperparameters or what are good architecture choices? And here we show that, for example, if you look at the architectural choice, 3D-UNet versus other architectures, you can see that, well, okay, at least the best performing methods all use 3D-UNet-like architectures. But at the same time, 3D unit is also scattered throughout the rankings. So there's also people that use 3D unit and they got last ranking here. So it's not a guarantee for success, right? And if you go one step more detailed and you look at different architectural variations, you can see that this holds true basically for any thing that you look at. So there's no guarantee. It might work, it might not work, and anything can work. So that means if you have your own data set now, you're not smarter at all. You need basically to start from scratch and think about, okay, what am I going to do to solve this task now without any guidance? Going even one step further here and only looking at one architectural variation, you can see that the hyperparameters are varying, right? So there, if we now look at the hyperparameters of this similar method configuration, so the rankings vary from participant to participant, but also the parameterization of that method varies a lot. So it's a highly complex thing, and it's really unclear what to learn from such a challenge. And this is only one data set here, right? Kids challenge. And now in the biomedical domain, we have many, many different data sets. And here you can see an overview of different data sets, and you can see parameters that describe the data set as a little chart. And you can see that the parameters vary between data sets. They vary a lot. So the median number of voxels, the size anisotropy, the spacing anisotropy, everything varies. So from data set to data set, we are facing very different challenges. So in summary, we can say that deep learning pipeline design is highly complex and imaging properties vary drastically. So we have to design our pipeline for each problem, and it's really hard to do. And an unit is designed to automate this. And in principle, we have a data fingerprint that basically derives some properties of the task. And then we have a set of parameters. Some are fixed, others are adapted by rules. And then we have also a remaining set of empirical parameters that we then optimize during the training process. And the good thing is, while it looks complex, it's completely automatic. So you just plug in basically your data set and a unit configures itself. So the development and optimization of the parameter settings has been done on 10 data sets. So we learned from 10 data sets what is a good configuration. And then we have used 13 additional data sets to basically test whether we were successful. And here you can see some results. And you can see that an unit handles a wide variety of structures already, many different image properties, so organs or cells or lesions. Modalities are CT, MRI, microscopy imaging, anything. So it's really adapting itself to very heterogeneous tasks. And yeah, we have been very successful also in competing with NN unit. So here you can see a list of tasks that we encountered in different challenges, and you can also see, so these are international challenges, and NN unit competed against specialized teams, right, that tried to optimize their method on the task. We just plugged in NN unit and it optimized itself, and it won 33 out of 53 tasks. And you can also see the red dot is NN unit. In case we didn't win, we were still in the top performing area of the leaderboards. So this has been really successful. But it's of course also only semantic segmentation, right? I've talked about this already. And at the moment, we are also trying to adapt similar methods to object detection. We call it NN detection. So NN in NN unit or in NN detection stands for no new. And that's because the NN unit is basically not a new unit, but it's a known unit which is configured automatically. Similar now here for detection. Following the NN unit agenda, we are trying to find a detection method that generalizes to many domains and tasks. And the problem here is that if you're trying to find a detection method that actually works for many, many tasks, you need many tasks, right? And it's not really easy to find many tasks for detection. And it's also not easy to find methods that you can compare yourself against, because as far as I know, there is no method that can just handle 10 different data sets at the same time. So we needed, as a baseline, we needed something that can handle different data sets. So we adapted NNUnit, used NNUnit basically as a baseline. So we ran the segmentation of NNUnit, and then what you receive is a pixel-wise map, and then you have to somehow find heuristics to come to an object level. So you can say, okay, majority of voxels decides, or whatever. You have some simple rule to go to object level, and then you can draw a box around your segmentation, and you have a detection output. So this is NNUnit basic, and we also have NNUnit plus, where we basically empirically optimize these decisions of how to come from voxel level to object level. So here's the design overview. It looks a little bit similar to NNUnit, but of course there are different parameters to optimize. I'm not going to go into too much detail here. And here are some results. So in green, you can see NN detection. And in blue, you can see the NNUnit plus, which is optimized empirically, and the NNUnit basic, where we just have some manual heuristics to go from voxel level to object level. Yeah, and you can also see that we have a large number of data sets here. And these data sets actually did not exist before. So many challenges are working on segmentation, as I've told you before. So what we did is we adapted many of those segmentation challenges to become basically object detection benchmarks here by just redefining the aim or the metric. And yeah, what you can also see is that the NN detection framework beats the basic NNUnit in 11 out of 12 tasks. And if you compare NN detection to the NNUnit plus in blue, you can see that we still beat the NNUnit plus in 8 out of 12 tasks. We have also ran NN detection on one of the few existing detection challenges. And you can see that we were able to retrieve the best result here. If you take out the two methods that are shown on the right, that include a step of false positive reduction. So we did decide not to use any of these false positive reduction steps because it's kind of out of scope for NN detection as it requires manual intervention. Yeah, and here I got a little sneak preview of NN detection version 2. This is not the final word here because it's only a single fold that I'm showing you. But it is a comparison of the version 1 that I've shown you before, and version 2 outperforms version 1 on many data sets. And so really looking forward to seeing the full results here on NN detection version 2. All right, so this was generalization. So my last words or my last part is now talking about representations. So what can we say about representations that we are learning, and what are some of the ideas that we are following at the moment? And one example I wanted to give you is here our work on multi-class segmentation. So I think that instead of going here from the partially labeled data sets in the middle, so we have many different data sets, and typically data sets are created to solve one problem, right? So we have a data set where the liver is segmented, and then we have another data set where the lung was interesting and we have something in the lung segmented, and we have a third data set and there's only the pancreas segmented. So we have these data sets that are partially labeled, and typically we would go here from the middle to the left. So for each data set we would train one model, one representation that is able to solve exactly that task. And I think that's not really efficient. So I think it in the future would make sense to build models that are able to handle really multiple tasks and that are able to aggregate knowledge and to basically profit also from the knowledge from other tasks in solving the task at hand. So we are at the moment going into the direction of being able to combine all those partially labeled data sets and creating one model, a multi-class model, that then predicts anything that it knows. So in order to achieve that, we combined here 13 public data sets, and these included 47 partially overlapping classes. And for each of these 47 classes, we basically trained individual heads for segmenting these structures, but using the same deep learning model. Here you can see some results. So you can see that the average DICE and also HD95 and MSD, we are all competitive basically. And in the cases where other methods beat us, it's only by a very small margin. So it worked quite nicely. And I think these are only some preliminary results, and they already show that the idea that we're implementing here outperforms related methods. So there are other related methods that go into the same direction of learning many different tasks at the same time. We are also able to match the NN-UNet performance on each of the individual data sets, which is important, I guess, because you wouldn't be wanting to sacrifice performance on any of the tasks. And yeah, we could also show that we match the state of the art in 3 public challenges. So this has a couple of advantages. It's basically very fast. So you have one model, you run it once and you get anything you want, instead of running individual models, which involve a lot of computation and is just not as efficient. And it also shows a great potential for transfer learning in the future, because you actually combine the knowledge from very many different tasks here in one network. So a second example I wanted to give you that is revolving around the representation is this one here. So I think in medical image computing, we are always dealing with radiologic, in my division at least, we are always dealing with radiologic images, most of the times at least. And usually what happens is patients go to the doctor several times. So we have time series of data. And our models are able to nicely handle 3D, they are able to nicely handle 4D, and they are even nicely able to handle time series data if the time series is equidistant, like in a video. But what we have here is we have another situation usually. We have data sets that were acquired at different points in time, and the time points that we use to acquire them are scarce. So we only have 8 acquisitions, for example, and they are at some arbitrary positions in time. And our current models do not have the ability to cope with such time series. So how can we learn from sparse and irregular time series? I mean, there are a couple of methods that you might be aware of. Gaussian processes, for example, are one choice here. However, these are rather suited for scalar function approximations, and neural processes are much better suited here. They are deterministic or probabilistic, as you might want it for your problem. And yeah, they can encode in principle anything, including images. So our idea is here to merge the neural processes with a UNet architecture, and show you some results on that. So here you can see a reference movement of 2 objects which are flying around. So this is basically the underlying biological process which is going on, right? But then you have a context, and the context is what we call is the view you get basically on your data. So you have a couple of scarce time shots, which you can see here on the left. So only 3 or 4 time points are taken, and then you want to basically interpolate between these time points in your prediction, also extrapolate. And you can see that it works really nicely here with our approach. For these examples, of course, there's other cases which fail. For example, here you can see on the top that the dot basically disappears, or you can see that the rotation of the star is not actually captured by the model because there's not enough information here available. So how does the model look like? So classically, what you would do in order to model time evolution of a disease is you would have some kind of a biological model. But here we are trying to learn it from data now. So we model here the tumor growth of brain tumor patients that are represented by some time points on the time axis. It's around 400 patients here that we are using. We are modeling this tumor growth as a probabilistic segmentation problem. And we are predicting the distribution of possible future segmentations here. In order to do so, we basically encode the different time steps using an encoder on the left side here. And then we have the typical sum of these encodings, as we usually do that in neural processes. And we also have introduced here some attention mechanisms in this method. And in the end, you are basically able then to sample from this global representation that you're creating, sample from it and pick any time you desire and make a prediction for that time. So here you can see for the reference shown in red, some mean predictions and also some samples of our model. You can see that we do get some variation between our different predictions here, which all look kind of realistic, so to say. It's really hard to of course, to say this is really realistic because we don't have a ground truth for all the potential possibilities that would be there for one patient. But you can also see that a neural process, for example, does not represent this variability that we know that exists. So it's actually very hard to predict what will actually happen. And we don't care about the correct prediction. So it does not need to be correct, but it should be realistic and it should show the diversity that we are actually seeing in reality. So we are using also quantitative measures to evaluate our methods. For example, you can evaluate the surprise. So how much surprise do I see when a model encounters a real patient development? And the nice thing now is that all this that we have shown to be able to model tumor growth is not specific to tumor growth. So you could use this model to model any stochastic image time series or any disease. It's just learning the disease progression from data, which I think is really a nice way to think about medical image computing problems in the future, because actually all of our data looks like this, right? We are only taking one point in time, but if you think about the patient, usually you have sparse time series, which is irregularly sampled. And I think we should start thinking about how we can make use of these time series. All right, so I've talked about 4 different problem areas in my talk. Talked about data and federated systems. I talked about solving the right problem and choosing the right metric. I've also talked about generalization between data sets. How can we make sure that our methods do not only work on our data set that we used for development? And I've talked about representations and some of our ideas that we have in that direction. And with this, I'm already at my end, and would like to thank my collaborators and, of course, the group that made this possible. So these people did the work that I was showing. So thanks a lot. Also, thanks to you for listening, and looking forward to questions if there may be any.
Thank you so much, Klaus, and thanks to the whole group, and thank you for sharing their work. We have time for, I think, 2, maybe 3 questions. Yeah, there are a few good ones here. Start with this one from Dimitro. So about the multi-headed network, the multi-class learning. how did you handle the diversity of the data sets, such as various contrast phases, different reconstruction kernels, etc.? Is it as straightforward as combining everything together, or do you need specific methods to handle basically different types of input data?
Yeah, very good question. Thank you. Indeed, we are handling the data set properties, reconstruction kernels, and anything we are handling just by providing them to our model and learning from it. So we don't do anything specific to take care of that. The only thing is so even if you look at one structure, for example, the liver, and you have 3 different data sets that include liver segmentations, you might be facing different definitions of what is liver and what not. So what we do is we are not throwing all of these definitions into one, but we are learning different heads, and then we are basically trying at post-processing to make sure that we are combining those different definitions in a manner that we would like to see. So this is the only thing that we basically do in order to get rid of systematic difference between data sets.
So basically, some of the heads may have, during training, only seen data coming from specific, call it submodalities.
Yes.
Mm-hmm. Okay.
What we are also doing, I forgot, is so some data sets do not show the whole body.
Mm-hmm.
So if you have maybe one head which is trained on segmenting the liver, and the images are always focused around the liver, and then during test time you throw in a data set that only shows the lung, then you might get into trouble, because the model basically is confronted with data which is not which is basically out of distribution, right? And then it's very hard to predict what's going to happen. So we do have a step that is estimating the position. So basically the slice indices that are shown in the image, and then we mask out predictions that are out of distribution.
Mm-hmm. Nice question from Chris on a completely different aspect. What is the current state of the regulatory and the privacy requirements for federated training data? Right? Is the ecosystem sort of right for actually being able to utilize this bottom of the iceberg component of the possible available training data sets?
Yeah. Thank you for this question. This is, of course, something you are confronted with as soon as you start thinking about federated scenarios. And even though I'm working in this area for quite a long time now, I'm still not an expert in all the different aspects. And what I can tell you is that Germany is probably one of the most difficult examples if you talk about these regulatory aspects and privacy issues, because it's really, really strict on the one hand compared to other countries, and it's also federated. So Germany is also in itself federated. So we have different states, and each state has basically its own law when it comes to privacy protection. And each state has also its own privacy protection officer. So basically, if we are building now up our network of 36 clinics in Germany, we need to talk to many, many different privacy officers in different states and try to come up with a compromise. And I can assure you, if you have basically 13 goes from 15 privacy officers, there are still going to be 2 that say, no, this is not the way we want it. But there are some there's some movement also on the regulatory part. So there are, for example, some new laws in Germany that now allow focusing only on one state, and then there's negotiations how the other states then are going to accept or not accept things. So I mean, things are moving, but it's still, of course, a challenge. So, and my advice would be in such large-scale consortia to not try to do that all on your own, but you have to have some kind of expertise in your team to solve these things. And yeah, as a computer scientist, I'm actually not the right person to talk to to talk to the people that take charge. But it's of course my responsibility to explain what our methods do, and that also our methods are coherent basically with the regulatory aspects. So yeah, there are some big challenges ahead, but I'm convinced that it makes sense to take this route and to go this way. And I'm also convinced that we are going to solve these hurdles, even though if it's hard at the moment. There's going to be more and more progress, because the really grand challenges in the field, they can only be solved together. And I doubt that we are always going to agree on sending all data from the world to one location in order to solve a problem. That's just not going to be the case. So I think we need to solve it.
Well, let's hope so. Yeah, for everyone's sake. Did I understand correctly? The challenge you mentioned, the grand challenge, I think it was the Bratz test. So are you doing the federated data? Are you using that just for validation or also for training? Or you're just validating?
The validation.
So validation. So that ties into these issues of getting access for training.
So that makes it a little bit easier, because you can exactly control what happens, right? So what we are doing is we are packaging our methods basically in containers, and we are negotiating with each partner in the network about security aspects of those containers, and then how they make sure that those containers are internally in the clinic run in an environment that does not allow any breakout. And then we make sure that the data that is allowed to be used in the challenge is also in that secured environment in that clinic, and we also make sure that there's nothing sent by the method to any
outside
party outside. Yes, and of course there's a little bit of trust also involved, because we during the registration of participants, we can also run background checks. Is it really a scientific institution here? Is there really some interest in the challenge, and that kind of things? And these also help to build up the trust that you need in order to let methods into such an environment.
Sounds like a big operation to get this off the ground. Yeah. Okay, thank you very much. Yeah, I wish we had time for a few more questions, but we need to wrap things up. So thank you again, Klaus. It was a pleasure having you here. Thanks again for everybody for participating, and please feel free to reach out to either of us over LinkedIn or email, etc. If you have further questions, and hope to see you all next time at the next webinar. So thank you again, and yeah, have a great rest of your day.
Thanks a lot for the invitation. Have a great day too.
Nice chatting with you. Bye.
Bye.